Hello, my name is Martin Frejno, and I would like to thank the organizing committee of the Computational Mass Spectrometry Community of Special Interest for giving me the chance to present to you an AI-driven leap forward in peptide identification through the deconvolution of chimeric spectra. As most of you will know, traditional search engines usually largely disregard the intensity dimension of experimental mass spectra. Instead, what they do is they calculate all possible theoretical fragment ions for a given peptide candidate, assign them unit intensities, and compare how many of those fragment ions do they actually observe in the experimental spectrum. However, what one would actually want to quantify is not only the presence and absence of fragment ions, but also their intensities, since the fragmentation behavior of a given peptide is a function of its sequence, its charge, and the collision energy used for the acquisition of the experimental spectrum. Traditionally, these intensities originated from experimental spectral libraries that took actually quite some time to generate, but nowadays they can be readily predicted using deep learning. We at MSAID also generated a deep learning framework called Inferis that utilizes transformer architecture and was in fact already incorporated in Proteome Discoverer 2.5. Back in 2020, it could predict the retention time and fragmentation behavior of triptych peptides that were acquired using HCD. But this year, we diversified our training dataset and can now also support CID fragmentation, as well as TMT and TMT Pro labeled peptides, and also substantially increased our prediction accuracy for non-triptych peptides and singly charged analytes. This is shown in the plot on the right, which visualizes distributions of normalized spectral contrast angle as a function of precursor charge for Inferis 1.0 in orange and Inferis 2.0 in blue. As you can see, Inferis 2.0 consistently outperforms Inferis 1.0 in all charge states, but in particular does so for charge 1 peptides. With these accurate predictions in hand, we set out to tackle one of the fundamental problems in bottom-up mass spectrometry-based proteomics, and that is, of course, chimeric spectra. But to us, the extent of that problem really only became clear when we looked at the human spectral library consisting of roughly 400,000 precursors and visualized the number of precursors as a function of retention time and precursor MZ. We binned that space in 2 Thomson by 1 minute isolation windows, and if you place such an isolation window anywhere in this space, you will nearly always pick multiple peptides for fragmentation. And that is of course not even considering DIA type of acquisition methods, which by design violate the central dogma of mass spectrometry-based proteomics that one spectrum produces one peptide ID. On the foundation of our predictions, we devised an AI-driven and spectrum-centric algorithm for the deconvolution of chimeric spectra. In essence, it tries to maximize the explained intensity with a minimal set of peptides and also calculates the contributions of these peptides to the experimentally observed total ion chromatogram. Those contributions can later on be used for quantification. All this happens in a single concerted step where all peptide candidates from the entire isolation window compete against one another for experimental intensity. That is visualized in the plot here, where we have the experimental intensity of a typical MS2 scan below the x-axis, and now up top we see the first, the second, the third, and the fourth identification produced by our algorithm. As you can see, the predicted intensities in colors on top of the x-axis correspond very well to the experimental intensities below the x-axis, and the predicted retention times suggest that these peptides should co-elute. And if we now check in the MS1 isolation window that was used to trigger that MS2 spectrum, we see that all 4 precursors are present and are in fact co-eluting. This algorithm is termed CHIMERYS and is integrated into Proteome Discoverer 3.0. CHIMERYS essentially consists of 5 different steps. We start out with a prediction of peptide properties, as I alluded to earlier, and then perform a first search in order to generate a couple 10,000 PSMs without considering retention time as a figure of merit. These PSMs are then used to recalibrate both our retention time and fragment ion intensity models before we enter our main search that also performs deconvolution of chimeric spectra. FDR in the end is then estimated using percolator. With chimeras, we can essentially double the number of peptide identifications in complex proteomes that were acquired using DDA. This is shown in the plot here on the left, which visualizes the number of PSMs, peptides, and protein groups identified using chimeras in orange, or previous versions of Proteome Discoverer utilizing sequest. As you can see, chimeras nearly quintuples the number of PSMs, nearly doubles the number of peptides, and adds 50% more protein groups in comparison to sequest HT. And what is very encouraging is that chimeras identifies nearly all peptides identified by previous versions of Proteome Discoverer utilizing sequest and adds additional identifications on top of that. However, chimeras not only works for complex full proteomes, but also highly fractionated samples, as the one shown here on the left. Here I visualize the number of PSMs, peptides, and protein groups identified by chimeras in orange and sequest HT in gray from the Arabidopsis thaliana deep proteome that is part of the Kingdoms of Life dataset. Even in this highly fractionated scenario, chimeras was still able to increase the number of identified peptides by 38% compared to sequest HT. However, if you are not interested in digging deep into your sample, but are instead interested in high throughput experiments with chimeras, you can now run your chromatography 3 times as fast and still retain the same number of identifications with chimeras as you were previously able to obtain using sequest HT. This is shown in the plot on the right, where I visualize the number of identified peptides as a function of gradient length. But what is really cool is that because we can now deconvolute chimeric spectra, we can adapt our measurement to our data analysis and really acquire data using wide isolation windows or wide window DDA. And if you do that, you get the plot here on the left, where I visualize the number of identified PSMs as a function of isolation width. And when we ramped the isolation width from 0.4 to 8 Thomson, what you see is that when using chimeras, the number of PSMs rapidly rise and then start to saturate once you reach about an isolation window of 4 to 5 Thomson. However, if you do use isolation windows that wide, you want to make sure that the additional candidates that you need to consider for the scoring of each spectrum don't mess with your error control. And in order to check that, we performed entrapment experiments, searching basically 4 different isolation windows of a human sample against a human FASTA file, concatenated to 9 copies of itself, where we shuffled peptides at the peptide level. And then we basically plot the normal target decoy FDR on the x-axis and also calculate an entrapment FDR based on the matches into shuffled peptides and plot that on the y-axis. In these types of plots, a well-calibrated scoring function basically means that your curve is very close to the diagonal. However, for sequest HT with precursor detector, that is a search engine dedicated for the analysis of chimeric spectra, we see that as we increase the isolation window, we basically increase the error that we make in the sense that sequest becomes more and more anti-conservative the bigger the isolation window gets. However, that is in fact not the case for chimeras, as visualized here on the right, because it doesn't really matter whether you acquired the data with 0.4 or 8 Thomson isolation window. All of these curves basically overlap with one another and are very close to the diagonal. What I've shown you until now is the current state of chimeras as it exists in Proteome Discoverer 3.0. And now I want to switch gears a bit and talk about the future of chimeras, and in particular, basically about the meaning of the last slide, because that begs the question, do we really have to choose between DDA and DIA? Are the two actually that different, or are they in fact the same? But before we dive deeper into this, I want to remind you of how DIA data is typically analyzed, and that is using a peptide-centric approach. This approach combines identification and quantification in a single step and essentially evaluates extracted ion chromatograms for both fragment ions and precursor ions. Our spectrum-centric approach, on the other hand, separates identification from quantification and performs identification using the currency that we all measure, the spectra, and quantification using the contributions that I mentioned earlier. Essentially, we calculate contributions for each peptide to its corresponding experimental spectra and trace those contributions across retention time to generate peptide-level quantification. In order to evaluate this approach, we analyzed a subset of the new LFQ bench dataset published this year, which contains both DDA and DIA measurements of the same samples. We analyzed these samples with both MaxQuant and CHIMERYS and compared both identification and quantification performance. On the identification side, here on the left, you see that the overlap between MaxQuant and CHIMERYS is pretty high, which is very encouraging. But you'll also notice that CHIMERYS identifies many more peptides in the DDA file than in the DIA file. However, what is very encouraging is the fact that the CHIMERYS MS2-based quantification in the scatter plot on the right on the x-axis on the DIA file corresponds very well to the MaxQuant MS1-based quantification on the DDA file here on the y-axis. However, what was a bit more interesting for us at the time was a head-to-head comparison between CHIMERYS and DIAN on the DIA file. On the identification side of things, here on the left, you see that there's a big overlap between CHIMERYS and DIAN, which is very encouraging. And on the quantification side of things, here on the right, you see that the CHIMERYS MS2-based quantification on the x-axis corresponds very nicely to the DIAN MS2-based quantification on the y-axis. The quantification scatter plot is not centered around the diagonal, as it was the case for CHIMERYS and MaxQuant on the previous slide, but that just means that CHIMERYS and DIAN measure on slightly different scales. Next, we wanted to take a closer look at the 18,000 unique PCMs that were only identified by DIAN. In order to do that, we pass these PCMs through what we call the CHIMERYS trace mode, which basically quantifies where in the CHIMERYS workflow a given PSM is disregarded. CHIMERYS consists of several filter steps prior to percolator, which help to weed out shaky IDs early. And when we do that for PSMs that belong to DIAN unique PCMs, we find that for 20% of those, CHIMERYS was not able to find the most abundant peak of the prediction in the experimental spectrum. And for an additional 20%, CHIMERYS was not able to find the top 2 out of the top 3 most abundant peaks of the prediction in the experimental spectrum. And that is really puzzling, because you would expect that the most abundant fragment ions of a given peptide would show up out of the grass first. Additional 40% of the DIAN unique PSMs were then disregarded because they had too little contribution to the experimental spectrum, and another 20% of those PSMs were filtered out by percolator. We also performed entrapment analyses like before, but now at the PCM level, which by the way corresponds to peptide charge modification or precursor. And if we look at the precursor level entrapment curve for CHIMERYS in this DIA setting here, what we see is that just like for wide window DDA, we can properly control FDR visualized by the fact that the CHIMERYS entrapment curve is very close to the diagonal. I should mention that this is, of course, a very synthetic setting. We work here with a 9-fold shuffled entrapment database. Those sequences are very similar to the target sequences, but in this particular scenario, DIAN seems to be a bit anti-conservative and hence underestimates FDR in this setting. But now I want to switch gears a bit and talk about the intersection between DIAN and CHIMERYS, and in particular the most abundant peptide. I brought you two XICs here. DIAN is on the left, CHIMERYS is on the right, and you see that the experimental intensities will of course Those correspond 100% to one another. They are the same intensities. But for chimeras, we included a separate second dimension, which is below the x-axis. And that is basically the predicted intensity of each fragment ion scaled by the contribution of that peptide in that particular scan. And you can see if we plot it that way, you see that the prediction and the experiment correlate with one another across retention time, allowing us to do both identification and quantification as well. Another acquisition method frequently analyzed using XICs is of course PRM, and since CHIMERYS works nicely for DDA and DIA data, we hypothesized that it should also work nicely for PRM data. So we set out and did a HeLa DDA run, and from that run we selected 52 peptides from 18 proteins across the entire intensity range and targeted them in a separate PRM run. And then we performed data analysis using either Skyline or CHIMERYS searching the data against a human FASTA file. And if we do that, funny enough, CHIMERYS is actually able to identify 2,000 additional peptides to the ones that were actually targeted, allowing us to do proper FDR control. And when we do what I told you earlier and trace these contributions of peptides over retention time, what we get is the plot here on the left for the window that was used to target the orange peptide. And of course, the orange peptide is identified, but so are 3 additional peptides that elute a bit later than the orange peptide. And the area under these curves is then used for quantification. And when you do that and basically compare that quantification to the manual expert performed Skyline quantification, you get the scatter plot here on the right, which I find particularly striking in that the CHIMERYS automatic quantification basically corresponds to the manual expert performed Skyline quantification with a correlation coefficient of 0.99. So all in all, I hope that I've convinced you that the spectrum-centric approach implemented in CHIMERYS can be used for the analysis of DDA, wide window DDA, DIA, and PRM data, and that the contributions calculated by CHIMERYS can in fact be used to perform MS2-based quantification. And I think it's fair to say that we can predict a bright future for ML-based applications. And with that, I'd like to thank the MSAID team that made all of this possible, as well as our advisors, Professor Wilhelm and Professor Küster, and the Proteome Discoverer software team. And if you have any questions, feel free to join our Discord server. That's the QR code for it, and follow us on Twitter and LinkedIn. And if you're one of those unicorns that can do both proteomics and bioinformatics, feel free to contact us because we are hiring. Thank you very much.